Thank you, Dr. Lynch. It's a pleasure to have the opportunity to share with you some of our recent results. Dr. Lynch has given you the title, and I'll move very quickly to point out that I have no disclosures regarding this research activity. And I'll start by stating the subject matter, which are epigenetic events that lead to instability of DNA repetitive elements during cancer initiation and progression. And these alterations occur at many thousands of genomic loci that harbor repetitive DNA elements. And these changes have a dramatic impact in cancer biology, which we don't yet understand very well, unfortunately. I'll also argue that monitoring the loss of epigenetic silencing of line 1 and other retroelements may have prognostic utility in cancer and may serve to define therapeutic outcomes in individual patients. Now, some teaching here, because this is a somewhat arcane subject for clinicians. It actually turns out that 47% of the genome is not comprised by genes, but actually by repetitive DNAs, either retrotransposons or DNA transposons, that have been added through the genome in evolution. And notably, a lot of these additions occurred in primates exclusively. Primates underwent something called the retrotransposon explosion, which added a lot of parts to the genome. And many of these parts were alternative promoters and enhancers and other controlling sequences that we don't yet understand very well because they're not present in mice, our favorite cancer model. Today, I will not be talking about all these families, but I will focus predominantly on line 1 elements, which are the subject of this presentation. But actually, all of these other families, ALUs, MIRs, SVAs, LTRs, endogenous retroviruses, do have roles to play in cancer, and there's literature supporting that assertion. So I'll point your attention to this paper, which was published in 2009 by a group in Japan, the Riken group, who used a technology called CAGE that maps transcriptional initiation sites. And looking at human tissues predominantly, not cancers, they identified approximately 23,000 candidate regulatory regions that were transcribed and were derived from retrotransposon sequences. And notably, the transcription of most of these loci was found to be tissue restricted. In fact, more tissue restricted than gene expression itself, which was surprising. The same authors have published a review in 2010 regarding transcriptional regulation in the mammalian genome. And they point out that the genome is pervasively transcribed to produce a variety of short and long non-protein coding RNAs, including many derived from retrotransposon sequences, which also play important roles in the epigenetic regulation of gene expression, particularly chromatin silencing states. So they finish by saying that the mammalian genome might be more accurately viewed as islands of protein coding information in a sea of cis and trans acting regulatory sequences, sort of like the Italian boat being the genes and the Mediterranean being all these transcripts, with the outcome that the genes get derailed.
Hopefully the outcome's better than the Italian ship.
Yes, not yet. So some examples of regulatory sequences that would be encoded in primate-specific members of repetitive DNAs include alternative promoters, alternative enhancers, which have been well documented, and trans-acting non-coding RNAs that have regulatory roles, of which relatively few are documented, but are known to exist and are very well documented in plants. Also, examples are oncoproteins derived from retrotransposons, which include reverse transcriptase, REC, and an endonuclease, the ORF2 endonuclease of the line 1 retrotransposon, encoded by line 1 elements, which are the second most abundant retrotransposons in the human genome. I don't have time to go over all of these examples, but I will focus on the ORF2 endonuclease as an oncoprotein by showing you a recent publication, 2009, from the group of Michael Rosenfeld. And this group showed that line 1 ORF2 endonuclease acting in concert with activation-induced deaminase synergistically generates double-strand breaks at juxtaposition translocation loci, specifically driven by androgen receptor binding events. What this means is that androgen receptor can bind to specific loci in the genome and drive the juxtaposition of these loci even if they're far away, many thousands of bases. And then through mechanisms that we don't quite understand, these junctures attract activation-induced deaminase and the ORF2 oncoprotein generated by line 1 transcripts. And together, these two endonucleases generate breaks that eventually lead to tumor translocations, which are not random, but they're driven by the androgen receptor. But these are recent findings, and this story has not been developed fully, so I have it here as a teaser. Now, going back to epigenetic instability, I would point out that DNA methylation alterations occur at many thousands of genomic loci, and they have a dramatic impact in cancer biology, as I said at the beginning. I've said it twice. And you're going to see in this talk how we are making attempts to monitor epigenetic silencing of line 1 elements using a system that is generic and extensible. And by looking at the epigenetic silencing of line 1 elements and the loss of silencing in cancer, we may be able to have prognostic tools and even tools that have utility in defining therapeutic outcomes in cancer patients. So it has been shown, this is a 2008 paper by a group at Harvard, that loss of line 1 methylation is independently associated with shorter survival among colon cancer patients. The study is quite large. It involved over 600 individuals. And the bottom line is that if you divide these individuals on whether they have normal line 1 methylation or low line 1 methylation, which is the green line, in the Kaplan-Meier curve, you observe much shorter survival for those patients that have the most dramatic loss of line 1 methylation. So line 1 is a marker for poor outcomes in colon cancer. More provocatively, the recent work from Japan, and this is January 2011, almost exactly a year ago, this work showed that if you look again at colon cancer patients and look at the response of patients to oral fluoropyrimidines, 5 of you in this case, and compare the outcome with surgery alone or using chemotherapy with the fluoropyrimidines, you find that for patients that have normal line 1 methylation, there's no difference between surgery and therapy. However, for patients with low line 1 methylation, that is with loss of silencing, the focus of the talk, loss of silencing. In these patients, you add the oral fluoropyrimidine, and you now get a much better outcome than surgery alone. So as I told you in the previous slides, the patients with low methylation have poor outcomes. In those patients, the drug has a stronger effect. How is that? Well, we don't quite understand, but the proposal is that there is a synergism between 5-fluorouridine treatment and line 1 transcription in a way that line 1 transcripts possibly induce more DNA breaks in the presence of the drug and cause the cells to die. So line 1 expression then in these patients is a bad thing. But when you add the drug, it's a good thing because this expression conspires with the drug to kill the cells. But again, these mechanisms are only beginning to be studied, these synergisms. So that's the introduction. The rest of the talk is intended to give you proof of concept of a novel imaging tool for observing epigenetic dysregulation events involving specific repetitive DNA elements in living cells. These imaging biosensors that we designed for studying live cell chromatin states in tissue in the context of disease and therapeutic responses have the following functional imaging attributes. First, they are sequence specific and can be designed at will to target loci that are chosen to be the loci of interest. Two, these biosensors are sensitive to changes in DNA accessibility, so if accessibility goes up, a light signal is generated. And third, the ability to interrogate living cells in real time is performed without activating stress responses, because we do not use the green fluorescent protein and we do not need laser illumination. This is a luciferase-driven system, so it would allow us to perform the studies without activating stress responses. So in order to design the system, we took advantage of work done at the Rockefeller University in the lab of Tom Muir. And Edmund Schwartz from that lab had published a paper a number of years ago showing that in living fruit flies, one could use the drug rapamycin to observe post-translational enzyme activation driven by this drug using an artificial biosensor system that reports a protein-protein interaction. And the way it works is that the drug can be bound by these rapamycin binding domains, but the rapamycin binding domains are tethered to inteins, which are proteins capable of undergoing protein splicing. And the way that they configured the inteins was such that if they added a half luciferase at the amino terminus and the other half of luciferase at the C terminus, upon proximity induced by rapamycin, the luciferase would be spliced and then would become active, and then you could see luciferase activity in the flies over time and for several days. So the advantages of this system are that you don't use green fluorescent proteins, so you don't activate stress responses. And B, that perhaps we could substitute rapamycin for something more interesting. And what we did was substitute rapamycin for DNA sequences. And what we're going to do is target a DNA sequence using proteins capable of binding specific sequences. And nature has had those at hand for millions of years. So we took advantage of polydactyl Zinc proteins, which recognize sequence-specific domains in double-stranded DNA via interactions through the major group in double-stranded DNA. So we can design zinc fingers to target a sequence at will. The way the system works then is analogous to the rapamycin-driven system. Zinc finger A binds at the sequence that we chose. Zinc finger B binds at this other sequence that we chose. It's tethered to intein, tethered to half a luciferase. Same story here. After binding, protein splicing of the inteins occur, generating active luciferase, and therefore light. Now, in order for light to be generated, the DNA sequence has to be accessible for zinc finger binding. If the DNA sequence is silenced, tightly coupled to silencing complexes involving DNA methylation and methyl binding proteins, the zinc fingers would not be able to bind, and you would not get light. We then did theoretical calculation of the single molecule photon flux for luciferases, and realized that the firefly luciferase used at Rockefeller is a fairly low yield luciferase. So unless we substituted this luciferase for a higher photon yield enzyme, we would have to target multiple copy sequences. rather than try to redesign the system to be more sensitive, we decided to use what we had and to target sequences that have 1,000 targets per cell. And I think that was a wise thing to do, and we're following the wisdom shown in 1969 by Mary Lou Perdue and Joe Gall at Yale. When they did the first in situ hybridization experiments, they didn't target a single gene, they target alpha satellite repeats, and they succeeded. And then in situ hybridization succeeded in showing in situ hybridization of single copy genes 10 years later. So we didn't want to wait 10 years. We decided to target multiple copies. So we targeted the line 1 PA2 sequence locus. Line 1 PA2 is a sequence that is specific for primates. It's only present in humans, chimps, and orangutan, gorillas. It's not present in the rhesus macaque monkey. It's a fairly recent addition to the genome. And it's closely related to the human-specific line 1HS, which is known to be transpositionally active. And PA2 contains, is present in roughly 3,000 full-length copies in the human genome. So we designed zinc fingers that could bind in two sequence domains, domain 1, domain 2. Zinc fingers bind, again, using tethered system of intane and luciferase, we can generate splicing. We designed the system so that the luciferase would remain tethered. In that system, then, we know that we would get one luciferase per event. We get a lower yield than in the other system, but here we know how many we get. So we get one luciferase per binary binding event of the biosensor. We then tested the specificity of the zinc fingers that we designed to make sure that they were sequence specific. This is done by taking zinc finger 1, zinc finger 2, splitting this DNA, and then synthesizing oligos that represent either target 1 or target 2 alone, and doing an ELISA. And in that ELISA, tethering zinc fingers to maltose binding protein and measuring signal, which is essentially defined by the binding of maltose binding protein. The most important result is, of course, there's no protein binding in the absence of zinc fingers here. And the binding is dramatically reduced by even a single base change. So this is perfect binding for zinc finger 1. And this is a mutation of a single base in sequence 1, and you get much less binding. So we confirmed that any changes we made, 1, 2, 3 bases, would significantly abolish binding. And remember that to get a signal, you need 2 binding events. So this is kind of not additive, but it's sort of multiplicative requirement for specificity. We then performed assays with transfected artificial DNA targets. So using cultured HeLa cells, we transfected plasmids that contain the complete DNA target, that is the sequence at the bottom in a plasmid, mimicking line 1 PA2 retro element sequences. And the HeLa cells do contain these retro elements, but most of them are methylated, so they're not accessible, while the plasmid, being unmethylated upon transfection, should be at least partially accessible, although it could be partially methylated over time. So I have to go quickly because Narendra is coming after me, so I'll just focus on this slide. First, I'll tell you that the protein expression levels are in the range of module 1, 56,000 to 134,000 molecules per nucleus. And actually, it took me 6 months to persuade the postdocs to get me these numbers. Really wanted to know how much was being expressed. And to give you a ballpark figure, the amount of NF-kappa B in a cell nucleus is roughly 100,000 molecules. So we have as many proteins of biosensor modules as you have of NF-kappa B transcription factor in the mammalian cell. Then we add the DNA, which is the artificial target, and we get a signal. This is module 1 sequence, module 2 sequence. And this is the 2 modules, that is the biosensor modules, with an empty vector. So big problem here. Please notice that in the absence of the artificial in the absence of the artificial target, we get fairly strong signal. And only by putting in a whopping amount of 1200 nanograms, we get a readable signal with a deviation that's much higher than the background. So the system has background. It turns out the system used in Drosophila by the Rockefeller group also has background. So we knew we would have background. We didn't expect this much, but this is what we got. So we're really lucky that we're targeting something that there are 3,000 copies per nucleus. We then measured how many molecules per nucleus are actually transfected in this transfection, and the calculation is that we have transfected 1,900 molecules target here. So we need 1,900 molecules to get a really robust signal. But we're not worried because our target has 3,000 copies per cell, so it still might be detectable. So let's move on then to actual assays targeting chromosomal line 1PA2. So here, we're not going to introduce any artificial targets. What we're going to do is we're going to take the cells and we're going to treat them with a demethylating drug and wait a few days and see what happens. And we'll use dosages of demethylating drug that are the same dosages that you would find in the bloodstream of patients that are treated with these drugs. So these are clinically relevant doses of the drugs. We then look at the methylation of the line 1 PA2 elements by doing polymerase bisulfite polymerase chain reaction and epitoper sequencing for methylation. And we confirm that after 5 days, decitabine, which is one of the drugs we use, reduces methylation at least at 1, 2, 3, 4, 5, 6, 7 positions. So the methylation reduction is actually moderate, and the question is this enough to induce loss of silencing? These are the results with 2 different drugs. Using decitabine in concentrations ranging all the way to 2.5 micromolar, or 5-azacitidine ranging to 5 micromolar, again, clinically relevant dosages, we observe an increasing luciferase signal relative to the vanilla transfection control. We use vanilla to control for the amount of biosensors that each cell prep receives. So with 2 different drugs, then we are able to demonstrate a response that we believe is sequence-specific because we have shown that the fingers are sequence-specific and is drug dosage dependent. This is only a start, but I think we are on our track to having a workable biosensor system. So I'm getting close to the end here. A few more minutes. So we have preliminary proof of concept data for a modular imaging tool for observing epigenetic dysregulation in living human cells in real time. And our first generation biosensor reduced the loss of silencing of line 1 PA2 retroelements during treatment with 5-azacitidine or decitabine. We believe these assays will be applicable to high-throughput pharmaceutical screening for improved epigenetic modulation interventions. We have talked to friends in pharma, and they agree that these assays would be useful. The DGnA Biosensors would also be useful for research applications. We believe they're well suited for epi-IMAGING without subcellular resolution, of course, because luciferase is not going to tell us things happening in subcompartments. So this is for use in tissues, perhaps tissues growing in 3D matrices or small animal xenograft models. And for illustration, I'm showing you work from the lab of Thomas Weisbrock at Harvard, doing modeling of gliomas in 3D cultures, or work from the Weisletter lab at Mass General showing tumor xenografts. And we believe these kinds of models would be amenable to the use of these biosensors, which are genetically encoded and modular. So future experimental approaches enabled by epigenomic biosensors include, but are not limited to, the following. First, pharmacokinetic evaluations of demethylating drugs and histone deacetylase inhibitors. And to give an example, this was published a number of years ago from the group of Jean-Pierre Issa at MD Anderson Cancer Center, and they showed that in patients treated with low-dose decitabine for chronic myelogenous leukemia, and the resistance to methylate was related to very dramatic levels of loss of methylation. And then as treatment went by, this loss of methylation went up. And the way they interpret this data is they say that the patients that are resistant have lower line 1 methylation levels, but actually those cells are more susceptible to drugs. So over time, the cells are dying, and you have some kind of clonal selection. I don't have time to explain this data, which is complex. But basically, if we had our biosensors, we would be able to observe single cells, and we would be able to see which cells are dying and in what numbers, and we could do population modeling with real patient data from the blood of patients. Second application, the evaluation of the epigenetic stability of retroelement sequences in induced pluripotent stem cells, iPS cells, or cancer stem cells. This recent paper from the group of John Moran and Warner Green showed that upon reprogramming of somatic cells into iPS cells, there is activation of line 1 retroelement mobility, and that this mobility is greater in those cells that continue to express reprogramming factors for a long time. So there is modulation of line 1 mobility. We could avoid it if we could observe this in real time and change the iPS cell method and observe again the dynamics of this loss of epigenetic silencing. Another interesting application, and the one that interests me the most, is the evaluation of genetic instability and cancer risk in ex vivo 3D cultures of human cells derived from individual patients. And I've been talking about this with a number of people at Yale since 2008. We still don't have at Yale the systems that I would like to have to do this, but that's a subject for another day. But anyway, I borrowed these two figures from the web. This is a company called Perfecta 3D, which claims that they have some hanging drop cultures that facilitate generating spheroids, and that these are useful for personalized cancer treatment. So how would we use these? What we envision is that we could image patient-specific 3D cultures that harbor specific epigenomic biosensors. And we could look at those cultures in the context of each patient's genetic makeup that we would know by genotyping the patients. And using this ex vivo model, we could study chemopreventive as well as therapeutic strategies. So this is food for thought for the future. And my idea is that these cultures for each individual patient would give us individual cancer susceptibility architecture of the patient. And I'll end with a cute animation that Sebastian Spekowski did. So this is the biosensor recognizing a sequence. Here it's going to some tissue and glowing in some cells, but not in all cells, telling us that these cells have a problem with epigenetic stability, and maybe we should do something about it in a living patient. And I'll end by thanking my collaborators, Shudong Huang and Ram Narayaswamy, designed and implemented these biosensors. Collaborators, Derek Tumri at Yale for imaging applications, Pilar Blancafort at UNC Chapel Hill for zinc finger design, Tom Muir, Rockefeller University for the INTIN splicing system, Sebastian Spekowski, bioinformatics analysis, and help also from my colleagues in surgery and pathology, Clarence Sasaki and Jose Costa. Thank you.